Hi guys, so today we're going to be going through DNA replication, understanding why it's necessary, the enzymes and processes involved, and the applications of DNA replication in other biomedical areas. Before I start though, let's go over the structure of DNA and its nucleotides. So, as we know, DNA is made up of repeating units called nucleotides, because DNA is a polymer, meaning it's made up of several monomers, and these monomers are the nucleotides. The nucleotides have pentose sugars. In this case, this is a deoxyribose sugar for DNA. Then there is a phosphate group. And there is the nitrogenous bases, right? Now, these bases form hydrogen bonds with another base on another DNA strand, which results in what we know as the double helix structure. These are the hydrogen bonds, and the hydrogen bonds is what makes the double helix, or the entire DNA molecule rather, very stable. First of all, let's go over what DNA replication is exactly. So DNA replication is a process through which DNA strands or molecules are doubled to form exact copies with the same base sequence. And by base sequence, I mean the nitrogenous bases that we have along the double helix. Now, this is very important because DNA provides the code of all life forms on our planet. Right? Now, that allows for variation, and so all of us have to have DNA, and for that to happen, we have to preserve that genetic code by replication. So, yeah. Now, what are the uses for DNA replication? It's for reproduction and for growth and tissue repair. For reproduction, our genes are passed on from our parents to us, right? So that's why there's a need to pass on this genetic information to offspring. The reason we look so similar to our parents or our other family members is because that DNA containing genetic information has been replicated and thus passed down. It couldn't have been passed down without being replicated first. Okay? Then for growth and tissue repair, first of all, these processes that help with this are like mitosis or meiosis, and that involves, again, DNA replication before the splitting of the cells, which we'll find out in another video. That forms new cells which help organisms grow or replace dead or damaged cells. Now let's go into the exact base, complementary base sequence that DNA has. So as we know, these are two DNA strands, okay? They're complementary base strands. Let's say this is A, T, C, C, G. This has to be T, A, G, G, C, because adenine binds or hydrogen bonds with thymine, and cytosine hydrogen bonds with guanine. You need to know these names for the IB. Now, this allows for a high degree of accuracy when the base sequences are being copied for DNA replication. So let's say this double helix has been unwound and separated by enzymes we'll find out more about later. So there's A, T, C, C, G. And this must be T, A, G, G, C. So this is the original DNA strand, just more split apart because that is how replication is underwent. Now, from this, nucleotides are attached to these template strands. So these the double helix is split to form template strands for enzymes to come in and replicate this DNA, and it uses complementary bases and the theory of that to figure out the order of the nucleotides and copy to make an exact strand. So this is how the new bases would come in place and replicate the DNA. So from this, we can see that this is a new strand, this is a new strand. Now from one DNA strand, we've made 2 DNA strands. But what's important here is that each new DNA molecule has an original strand, this original strand, and it also has a new strand. So this is what you call semi-conservative nature. Let me just write that down for you guys. Semi-conservative. This type of replication specifically is called the semi-conservative nature of DNA replication, because from one DNA molecule, we get 2 DNA molecules because of replication, but those 2 DNA molecules each have one original and one new strand. This is very important. And again, this accuracy, the reason we can replicate these DNA strands so accurately is because of complementary bases. If we didn't have that theory, then how would we know that a thymine would bind with the adenine here as it did with the original strand, right? So, yeah, and then at the end, obviously, hydrogen bonds are added to stabilize the structure, like so, making 2 new molecules for you guys. Now, let's get into the actual enzymes that work. There's the helicase enzyme and DNA polymerase. Now, helicase is a ringed protein or enzyme that is involved in the separation of the DNA molecule. So it comes in, let's say this is the DNA molecule, helicase is working, and the helicase would be here, splitting up the DNA into this kind of thing so new nucleotides can attach, right? So one strand goes through the circle, the ring rather, and another strand goes around the ring to split it up, and it goes further and further to split it up. This is what you call the replication fork. So where the helicase is currently working at, like where that junction is between the 2 strands, that would be called a replication fork. We learn more about this later. Okay, now the next enzyme that comes through, this is like the heavy lifter, I guess you could say, is the DNA polymerase. This is the enzyme that actually assembles the DNA strand. So this assembles new DNA strands using free nucleotides. By free nucleotides, I mean just any nucleotides that are around in the area. The DNA polymerase picks those up and adds it to the ever-growing strand. Again, DNA polymerase requires that complementary base theory that I mentioned earlier in order to know which order the nucleotides have to go in. Okay, and yeah, that's pretty much it. Now let's move on to how DNA replication can be used outside of our bodies. So one thing we have to know about is the polymerase chain reaction. Or what we would know better as the PCR. This is the test we would all take during COVID or the coronavirus epidemic in order to test for if we have the virus. So now we're going to know how it works. This reaction involves a machine or like machinery called the thermocycler. You don't really need to know how it works. We're going to go through the process now, but this is just a type of lab machinery that we use to for polymerase chain reaction or PCR. And it's involved in the amplification of a target DNA sequence. What I mean by that is that the PCR will take a small strand of DNA, a small DNA molecule, a small sequence of DNA, and amplify it or multiply, or like, how would I say it? It basically makes copies of that DNA strand to make millions of copies, and that is what you call amplification. Now, there are three main components in the PCR. They're the primers, there's Taq polymerase, and there is the free nucleotides. Now, the primers are essentially just single-stranded polymers, again, of a short chain of nucleotides. Stranded polymers with nucleotides. But these, they contain nucleotides that are complementary to the target DNA. Again, utilizing that same theory of complementary base sequences. With these complementary nucleotides, it allows for a starting point for the actual DNA synthesis process to take place within PCR. Now, this is the important enzyme, Taq polymerase. It's very similar to DNA polymerase that we have, except it comes from a bacterium that lives in hot springs. Because of this, it's able to withstand high temperatures and not get denatured as a typical body enzyme would. Because in the PCR, as we come to learn, there's a lot of heating and reheating and cooling at very high temperatures. That's why we need something that is strong and won't get denatured. So this is the enzyme that's involved in producing DNA strands of the selected base sequence. Okay, now there are these free nucleotides. This is important because this is what the Taq polymerase actually uses to synthesize the new DNA strands. It's basically like the same free nucleotides we would have during DNA replication, as I mentioned earlier, except they're added in to the mixture. Now, let me tell you the step-by-step process. First of all, the components are mixed. Okay? Simply, it's in a tube, and it's put into the thermocycler. Then is denaturation. This is a key step and a keyword. Remember that, please. And that is when the mixture is heated. Like I said before, heated to very high temperatures, like 92 to 98. Now, this is not normal body temperatures as we would have. That's why DNA polymerase is not used, and a bacterium that could withstand it better is used, because otherwise DNA polymerase would get denatured, right? And this is in order to break hydrogen bonds. Not covalent bonds, though. Those are different. Because all this is doing is splitting the DNA strands up. So we have these strands of DNA. It just splits the hydrogen bonds in between, not the covalent bonds, because the covalent bonds would then break up the nucleotide itself. Okay? Then the third stage is annealing, another keyword. And this is when the mixture is actually cooled. And the primers bind to the lone DNA strands that have been split because of, or the lone RNA strands, depends, because of the hydrogen bonds breaking from the denaturation process before. Okay? And then the last stage is elongation. And this is when Taq polymerase comes into play. And it just catalyzes the synthesis of DNA. And also it uses the free nucleotides here. Now these steps are repeated over and over again until the target DNA length is produced, and the number of copies that are produced is required. So key things to remember here are these keywords: denatured, annealing, elongation, and the processes that take place. So this breaks hydrogen bonds, the primers bind here, and then Taq polymerase comes into play here with the free nucleotides. Okay? Now, the next process is gel electrophoresis. I've got this little diagram to show you guys. Basically, gel electrophoresis is, let me just write that down here. This separates DNA molecules by length, and it does so using a sheet of gel. So there's sheet of gel, roughly 3 to 4 millimeters long, thick. There's also an electrolyte solution, or buffer solution in this case, that's poured on top of it, and electrodes, as you can see in the diagram. Now let me explain the process. Basically, this gel sheet is placed in some kind of container with the electrolyte or buffer solution poured on top of it, and the gel has wells in it. You can't quite see clearly on here, but there are these wells made in which the DNA sample is pipetted. Now keep in mind, this is usually done for like unknown lengths and DNA, but one of the wells has to contain known lengths. So this is like the sample that we know. It has to have known lengths for us to compare what we get, or the results we get, so we know what the results actually are. Does that make sense? So what happens is a voltage is applied, and that results in the DNA moving from the negative electrode to the positive electrode. So it's negative to positive always. And that's because, which we'll find out in another video, DNA is negatively charged because of the phosphate group, so it moves away from negative to positive. And basically what happens is the longer DNA strands are therefore heavier. So because it's in the gel, there's greater resistance, and they're unable to move as far as a shorter, lighter DNA strand would move. So if the first arrow I drew was a shorter strand, another would go less, like a shorter distance, and that's a heavier DNA molecule, therefore it is a longer DNA molecule. So let me just make a note here. Short distance equals long molecule, and vice versa. You probably can't read that. Yeah, and that's pretty much it. I think of this similar to chromatography, in case you get confused, except with how chromatography works is that there are several inks placed, and a solvent helps these inks move a certain distance. The greater solubility, the greater the solvent will move. But in this case, it's how light, or I guess agile, that's not the correct word, but the easier they are to move, the lighter they are. So think of it like chromatography, you need a comparison sample as well to get your exact results. Now let's look at how these processes are used in the real world. So first of all, let's look at coronaviruses. So as I mentioned earlier, we used PCR frequently for our COVID testing, remember back in the 2020 pandemic. So it's a very real world issue. And for this one, a swab is taken, as I'm sure we all know about, and that's our DNA sample, okay? And in this, because it is viral and we have to we have to record the viral DNA strands, viral RNA strands, the RNA is converted to DNA using reverse transcriptase. Reverse transcriptase. This is an enzyme, again, and basically, I'll reiterate, the reason we convert the RNA to DNA is because the coronavirus has RNA as genetic material, and we have to convert that to DNA in order to get proper PCR results. Okay? Now, a PCR test is used to amplify these strands. However, these strands have fluorescent markers. Okay? And these markers are attached to any DNA with the viral sequence specifically. And the level of fluorescence is monitored, and once it reaches a threshold, that's how you know you have the virus or not. Okay? Now, the advantages to this is that it is very precise. It's specific, it can work with small quantities, so it's very helpful technology. However, some problems with that is that it's expensive. It requires specific lab equipment that's not very accessible, like the thermocycler, right? Yeah. Now the next example we're going to look at is DNA profiling. This is often used for paternity tests. And like forensic science or forensic medicine and all that. Like in crime scenes. So basically, again, a sample is taken from an individual, known or unknown, doesn't really matter. Then it goes through PCR in order to get the amplification, and then it undergoes electrophoresis, gel electrophoresis. Okay, now the pattern of DNA strands that are seen from the gel electrophoresis, the different lengths, is again compared to a sample that is, so we can see any matches between the individuals. However, in this specific testing though, there's something called short tandem repeats. Short tandem repeats. This is very important because this exact thing is what distinguishes between individuals. So in a genome, there are always 2 to 7 bases that are repeated, or consecutively repeated. Like, what I mean by this is that, maybe A, C, G, like C, this kind of sequence is repeated multiple times. A, C, G, C, A, C, G, C, right? This is repeated multiple times. However, the number of times this is repeated is what distinguishes between individuals. For example, my mom and I might have 4 repeats of ACGC. My mom might have 5 repeats of ACGC, but some random person that's not related to us might have 20 repeats of ACGC. So that this difference is what distinguishes between individuals. However, though, there's never 100% match, because it's highly unlikely that 2 people would have the exact same match. That's why my mom and I wouldn't have exactly 4, but like both of us won't both have 4 ACGC repeats. One would have 4, one would have 5, or there'd be some slight discrepancies. Okay? So that's pretty much it for the SL content. Anything further is HL, so continue watching if you're an HL student, please. Now I'm going to go more in depth. This is for the HL students, about the different, like the more other proteins involved and enzymes involved in DNA replication. So these are the new ones we're going to learn. We're going to learn about We're going to learn about DNA primase. We're going to learn about DNA polymerase 1. And we're going to learn about DNA polymerase 3. DNA ligase, and single stranded protein, binding protein. So DNA primase is something, it's like an enzyme that synthesizes an RNA primer. Okay? So basically what happens is that this RNA primer is released or synthesized by the DNA primase, and it marks the beginning of replication. So the DNA primase releases these RNA primers which bind to the template strand, and those primers signal to the DNA polymerases that this is where you have to start for DNA replication. Okay, now DNA polymerase 1 is the one that removes the RNA primer. Removes RNA primer. And then DNA polymerase 3 is the one we learned about earlier on. It's the major enzyme. It synthesizes a new strand. And it's also involved in proofreading, which we're going to learn about later. Now, DNA ligase is what joins DNA segments. Joins segments. And these segments called Okazaki fragments. I'm going to go more into that a bit later. Okazaki fragments. And then the single-stranded binding protein prevents H bonds from reforming. So these are the new ones we have to learn. DNA primase synthesizes RNA primers, which mark the beginning of replication. Then DNA polymerase comes along, removes RNA primers. DNA polymerase 3 synthesizes a new strand. DNA ligase is what joins any leftover segments and Okazaki fragments together, and single-stranded proteins prevent H bonds from binding. So now let's go from the beginning, what takes place step by step. First, helicase comes in, unzips the DNA molecule. Then it is DNA primase. Oh, sorry, not DNA primase. So after the helicase comes the single-stranded binding protein. That prevents any H bonds or hydrogen bonds from reforming, so it keeps the single strands separated. Then, is DNA primase. DNA primase comes in, creates the RNA primers. Then is DNA polymerase 1, which removes the RNA primers. Like, okay, I know we have to bind here. They remove it. Then DNA polymerase 3, which comes in, does the heavy lifting, actually synthesizes all those proteins. And then last but not least, DNA ligase comes along and seals up any any fragments that are disjoined. It joins the DNA molecules together and everything, and that's how you have your fully replicated DNA. Let's go back though on as to the different strands. We have something called leading strands and lagging strands. But what we have to know first is this is going to be talked more in depth in a later video, but we know that the DNA is made of these nucleotides. Hold on. They join like so. Right? Now, this is a pentose sugar, therefore it has 5 carbons. 1, 2, 3, 4, and then the fifth one's up here. 1, 2, 3, 4, and the fifth one's up here. 1, 2, 3, 4, and the fifth one is up here. And we can see that there's a 5 to 3 binding. This is what you call 5' to 3' binding. We'll go more in depth in this later on. So DNA always moves from a 5 to 3 direction, 5' to 3' direction. DNA polymerase also moves from a 5' to 3' direction. Okay, so this is very important. 5 to 3 going this way. So DNA polymerase will also move in this direction. Okay, now because the DNA strands are anti-parallel, again we're going to go in depth in this in another video. Anti-parallel basically means there are two DNA strands and they're parallel to each other, but one simply goes in the opposite direction. That DNA strand still goes 5 to 3, but just in the opposite direction, just flipped upside down. So if this one is 5' to 3', this strand would be 5' to 3'. Okay? Now this is very important when we distinguish between leading and lagging strands. So let's say this is a DNA. Okay? Let me just draw it out. I mentioned earlier on in this video something called a replication fork, and that's where the helicase enzyme is currently unfolding more. So let's say their helicase is working here, okay, at the moment. So this is what you'd call the replication fork. Let's say this strand is going 5' to 3'. Oh, it's not drawing. That means the other strand must go 3 to 5. Right? Now, DNA replication does occur in a 5 to 3 direction. Polymerase goes 5 to 3. That means, in this case, the DNA polymerase would be working in this direction because DNA strands are anti-parallel. So this strand over here, this is the DNA polymerase adding more nucleotides. This would go in a 5 to 3 direction. Okay? Now, because it's moving towards the replication fork, this is the leading strand. It's the leading strand because as the helicase unzips more and more of the strand, the template strand will be revealed, but the DNA primase has already dropped off some strands here, some RNA primers here. So the DNA polymerase 3 has already started replication here and will simply continue on as the helicase unzips more. However, the other strand that goes in the opposite direction is called the lagging strand. This is because the DNA polymerase has to move in a 5 to 3. That would be this way. 5' to 3'. Again, because of the anti-parallel nature. Because of that, the DNA primase has to start here. However, as more and more of the helicase enzyme unzips the protein, the DNA, more gets exposed, so the primase has to reattach some more, and then reattach some more, which causes some gaps, because as more of the primase and the RNA primers are attached, then the DNA polymerase has to restart, and they have to restart. They restart from here. They restart from here. But they have to continue in this direction. Because of that, these little fragments form, and these are what we call the Okazaki fragments. Okay, and so the leading strand is the leading strand because it's continuous and faster. The lagging strand is the lagging strand because it is slower and discontinuous. Let me just write these down for you. Quick, continuous. slower, discontinuous. It's discontinuous because of these Okazaki fragments, and then that's when the DNA ligase comes along and binds these up closer together. Now, one more thing we have to learn before we finish is DNA proofreading. Not DNA proofreading, just proofreading. This is carried out by DNA polymerase 3. Okay, and this is what makes DNA replication a very reliable and accurate process. Usually, errors are corrected as soon as possible in order to prevent mutations, because mutations, as we'll come to learn, are more often than not harmful. They're unlikely to be beneficial. And so if any mismatched nucleotides occur during replication, the mismatch is fixed as soon as possible, almost immediately, and that is done by DNA polymerase 3. So when, let's say, there's this, okay, oh wait, this is probably the wrong diagram to draw. I'm going to go back to the nucleotides. So let's say this is A, and then this is C, this is G, okay? But oh no, like this is 5 to 3 in this direction. So DNA polymerase must move in this direction as well. But as they come along, they realize C is incorrect. So what they do is they remove it from the third terminal before adding the next nucleotide. So it's realized, oh wait, this is wrong. So it's at A, it's realized C is wrong. So it removes it from here, removes it from here, removes it from here, removes it from here, and that is the only time the DNA polymerase 3 can move backwards. It moves backwards by one nucleotide to replace it, and then it continues moving forward. So this is the rare case it moves from 3' to 5', but that is purely for replacing and proofreading. Every other time it goes in a 5 to 3, and that is very, very important. And that is pretty much it for DNA replication. Hope you enjoyed and hope it helped.